Greetings, fellow scientists. I'd like to welcome you all to the Wisconsin Science Festival online field trip, A Day in the Life of a Scientist. Thank you so much for joining us today. We're ready to get started. My name is Dan from the Wisconsin Science Festival team, and we're here with special guests Aaron and Martha from the University of Wisconsin-Madison for this field trip experience. So how can you participate today? Well, you'll notice that your picture and audio are muted and the chat isn't available. But if you have any questions you'd like to ask, you can use the Q&A function at the bottom of your screen. As soon as you have a question, go ahead and type it in right away, and we'll ask the questions out loud during the field trip. This field trip will be recorded, and as always, we ask that you do this and all the activities of the Wisconsin Science Festival with a responsible adult. We'd also like to thank all of our sponsors and partners that help make the Wisconsin Science Festival possible. You can find out more activities and events during the festival all day today through the weekend and beyond at the wisconsinsciencefest.org. With that, we're excited to get started. So take it away, Martha and Aaron.
Well, I suppose I'll start. I think you've all seen a video of our lab. That's our general workspace. We're stem cell scientists, so we talked a little bit about what a stem cell is. But just to review, that's a specialized cell type in your body that can differentiate into almost every other cell type. And what we're particularly interested in is making cardiomyocytes. Those are the muscle cells of the heart, and they're the ones that are important for contraction. I'll turn it over to Aaron. We've got a few poll questions for you.
Martha, did you want to start by just going over a little bit of an intro of how you got to where you are?
All right. So I'm Martha. I'm a fourth-year graduate student in chemical and biological engineering. So I started as an undergrad in chemical engineering. Did a little research, got a position in a lab, mostly doing chores, cleaning things and doing menial tasks. Found that I really liked sort of the hands-on work and the creativity of lab work. And then I came here to Madison. Aaron and I are both in a five-year plan to graduate as PhD students.
Okay, thanks. And I'll give a little bit of my background. So I also got started doing research. I was a chemical engineering undergraduate, and I'm still a chemical engineering student now as well. So I started doing some research, I think, the end of my sophomore year. Although, even in high school, and I think even a little bit of middle school, there were a few opportunities where we were able to partake of some research opportunities. So I highly encourage that if you have those opportunities. It's a good way, even if you're not sure if it's something you're interested in, it's a good way to try and figure out if you're interested in something. So you're never going to know if you don't like something until you try it. So I'll kind of plug that there. So I had some different research experience in a lot of different stuff, astrophysics and things like that in like high school. And then started getting in my undergraduate degree, sophomore year, I started doing tissue engineering research, which is similar to kind of the research that I'm doing right now. I'm still working with a different type of stem cell and trying to make that become different cells that we were interested in. So I really liked the work that I was doing there, and by the time I ended up completing my degree, I decided I kind of wanted to stay on, and I was at the University of Oklahoma. So we actually had an accelerated program where you could get a master's degree in one additional year. So I decided I still wasn't quite sure if I wanted to go on and do a PhD yet, so I decided to enroll into the accelerated master's program, and so then in an additional year I got my master's. Towards the end of my master's, I decided I really liked the kind of the personal research and the kind of me being in charge of my own project and everything. So I decided to go on and start going towards my the full PhD. So I started applying, and then ended up here at University of Wisconsin, doing still kind of stem cell related research, where I'm at now.
Awesome. And where did you guys say that you were from? Are you from Wisconsin, or how did you end up in Madison?
I guess I can start with that. So the where you're from question is always a little bit more difficult. So I was born in Kansas and lived there for about 9 or 10 years. But then I did kind of my middle school and high school in Fort Worth, Texas. Hopefully I don't have too much of an accent, but if I do, okay. I've been told people can't usually tell where I'm from. But I lived there for a little bit before I ended up going to the University of Oklahoma. And there was kind of based on scholarships and things like that they'd offered me versus other schools is kind of why I ended up going there. They also had a pretty decently ranked program for chemical engineering, which is what I was interested in. Not that you have to know what you want to do whenever you go to college. Most people go into it and they're like, maybe I have a rough idea of what I want to do. But really the first couple of years and the kind of the intro classes you're taking, you kind of help to solidify if you're actually interested in that or if you want to transition to something else. There's a lot of opportunity for that. So I was kind of on the weirder end where I kind of knew going in I wanted to do chemical engineering because I had some exposure before. And then at the end of that, I applied to several different graduate school programs, predominantly in chemical engineering. And just kind of with the atmosphere and the collaborative nature of a lot of the research that's here on campus and kind of the atmosphere of Madison and everything as well is really kind of several of the factors that I took into account when I ended up deciding to come do the research here in our program.
Martha, how about you?
So I moved around too. I was born in Iowa. Most of my young career, elementary school, middle school was in Texas. Then my family moved to Minnesota for high school. And then I did my undergraduate at Kansas State, and now I'm here. So sort of bouncing around, mostly in the middle of the U.S.
And thinking about the day in the life of a scientist, if you could think about, we've got maybe some high schoolers joining us today, people in different interests in science. If you could think back to when you were in high school, did you think that a day in your life would be like what you're doing now? And how any like tips or advice you would give your past self about what a day in a life of your science life is like now?
So when I was in high school, my senior year, we created a first robotics group. So it's a competition team where you build a robot to complete tasks. And our task that year was to throw frisbees into a goal. So they had low, medium, and high heights. And we built different teams. So there were a group of mechanical people that kind of did the mechanical work. There were people that were an electrical team. And then we had sort of computer people, software engineers, I guess you would say. And that kind of gave people in my class a flavor of what they wanted to become. So sort of what the different types of engineering were. And we worked with people from companies in the town just to kind of see, what is it like in those different roles. So I got a feel for applied science is what I call it. So not just do something on a bench, but how do we make this thing actually work and throw frisbees? So from that, I really realized I want to be an engineering. So not just, let's try it on a bench in a tiny quantity. How do we make this big scale and actually feasible? So I started in general engineering, I guess is what you will say. So I just took the core engineering classes, physics, chemistry, calculus when I got to college and kind of decided from there what I wanted to go into.
Awesome. Aaron, how about you? Did you think that you would be doing today, a day in your life today when you were in high school, or how did you get to where you are today from there?
That's a good question. I guess so. Yeah, not at all that I think any of the stuff that I'm doing right now is at all related to any vision that I had when I was in high school. Partly because I wasn't, especially like pluripotent stem cells. I never knew I hadn't heard of those at all. I guess they were still, at least the induced pluripotent stem cells were still very new, I guess, at the time, if I go back to when those actually came out. So no idea at all. I didn't really even know that I wanted to do any bio work. And I had no idea that I was going to be doing bio. I mean, I enjoyed my biology classes and things like that. But as I mentioned, I kind of knew I wanted to go into chemical engineering. So it was kind of a divergence to start going onto the more the bio side of the research in the chemical engineering, just kind of using the chemical engineering mindset applied to some of those biological research questions. Yeah, I guess kind of I didn't really have any idea that that's, what I do now is at all what I would be doing. Some of the aspects are kind of similar, learning how to formulate a hypothesis and things like that are obviously things that we use every day, or at least on a long scale of, you generate a hypothesis and you can't test it right away. It might take you months or years to try to address that hypothesis. So the time scales are a lot longer than you might think. In doing some of your simpler high school biology experiments or any other experiments. And these are questions that aren't solved. So a lot of this stuff that you're doing in lab right now are all going to be, someone knows the answer. There is an expected outcome. Whereas this stuff right now is much more open space, much more free thinking. You can kind of use a lot of other literature and stuff that's out there and what is known to try to inform what you think might happen. But at the end of the day, you really have no idea what's going to happen. So it's much more exploratory, much more kind of deep thinking, and not just reading all the time. There is a lot of reading to understand kind of the context of what you're doing, but it's not like just here, read a lab manual and here, go do this experiment. Here's what the expected results should be. You're going to be graded on whether or not you get the correct result or not. So it's much more open, and I really enjoy that aspect of it. So being able to find out something that no one else knows.
With that, I think we can ask the audience a question now and get to at what kind of science you do day to day. So we're gonna send out a poll question related to the lab video that you saw. So did you watch that lab video before this meeting? And we'll see, that's a warm-up question, so we're gonna ask a few more questions during this field trip experience about the science that Aaron and Martha do. And we've got some questions coming in the Q&A, so keep those questions coming. So it looks like 3/4 of the people need a refresher on what you do in the lab. So can you paint a picture for us about, so you get up in the morning, and what do you do? Will you walk into a building, and what do you do when you start a day doing science? And what does it look like? Paint a picture for us.
Yeah, I guess I'll start with this one, and I'll let Martha fill in as well. So a lot of the work right now, because we are stem cell scientists, is taking care of the stem cells. So what we have to do is we have to feed them daily. So basically we have to give them fresh food every day. Which for cell culture is actually just a liquid. It's essentially, I think in Martha's, in the video, Martha mentioned, kind of like Powerade or Gatorade, essentially, that you're putting on your cells with all the nutrients that they need. That's basically the only thing they survive on. So a lot of the work is kind of changing that, what we call cell culture media, on the cells. We can add different things to that media to try to make the cells do what we want them to do. So sometimes you have to make a different media formulation and you're putting that on the cells. Right now, that takes up at least an hour or 2 hours of my day every day doing that work, because these are very, because I've got a lot of experiments going on. So I've got a lot of cells in there, a lot of stuff to take care of. When you're not in the lab, or when I'm not in the lab doing cell culture work, there's a lot of other kind of traditional chemistry or benchtop work as well. So if you're trying to run an assay, if you're trying to basically measure some aspect of the cells that you're working with, if you want to measure some specific chemical that they're producing, or if you want to measure some physical property of the cell, there's a bunch of other, what we call assays, to try to measure those different things. So performing some of those, and some of those might take on the order of an hour or so. Some of them might take several hours overnight, things like that. Or running on various different instruments. So I think also in the video, we showed a fancy microscope that we have set up. So a lot of work is actually taking pictures with the microscope to then analyze for things. And we can do that in many different modalities. So that's a pretty daily task, or at least weekly, we'll do some work on the microscope. And then I guess two other aspects of things that are kind of daily work are, one is reading papers. So, reading papers that other people have published that are related to your research to help inform your research. And so you're not duplicating efforts. If you design it, or if you think of it an experiment, if someone else has already done that experiment, it doesn't do you any good, and it doesn't do the field any good, or the general population any good, for you to spend more money to redo that same experiment. Sometimes there is use to that, because then you can replicate results, which goes to kind of one of the ideas in science is anything you present should be replicatable. Someone else should be able to do the same thing. But if you're trying to push in a novel direction and someone has already done that, it's more of a replication experiment and not as much of you're not going to be willing to devote as much time and resources to trying to just replicate someone else's results, unless you're going to then take another step. So reading is another big area of that. And then finally, kind of writing up or trying to synthesize your findings and distill them down into little bites that can be used. So either that's putting together presentations that we then give to other people all the time on new findings in our research, writing up papers, which is another big aspect of doing research, is writing up papers so that you can then share this with everyone on here's exactly what we did, here's how we did it, and here's our findings. So those are kind of the main aspects of kind of day-to-day work. I think one that I missed in there is actually analyzing the data. So you can do the assay and then you have to go and analyze the data as well, which is technically a separate part. So that's kind of those are kind of the main aspects of a day-to-day work. Martha, if I missed anything, feel free to let me know. Jump in here.
No, that sounds about right.
Pretty similar day for you, Martha. And you guys mentioned that you're working on cell therapies and taking care of cells. So maybe Martha, you could talk about like what do you do with these cells, and what kind of like how do you fit together your day-to-day with related to a question in the chat, what inspires you? So how do you connect, kind of how you do things every day to kind of these big questions that you have about science and your work?
Yeah, so that's a hard question I think everybody struggles with. So what's really important? I think for the field, the answer is the idea that we can take these induced pluripotent stem cells. So basically you can take a skin sample from somebody, make them into a pluripotent stem cell, which can differentiate into all cell types, and then differentiate those, say, into a cardiomyocyte. And then the idea is that you can use those cells for a cell therapy. So for example, after somebody gets a heart attack, those are usually caused by a blockage of your blood vessel. So if you can't get blood to the tissue, you're not getting nutrients, and then a lot of cells die. So the idea is that maybe we can provide them some stem cell-derived muscle cells to replace those cells that are dead and restore function to their heart. So this would reduce the need for transplants because, how do you get a large source of transplants? That's a problem in a lot of places. So it's starting in clinical trials actually in Japan. First in human has been successful. So the hope is, in my lifetime that this will be a real thing for people.
I guess what Martha mentioned there, a heart attack, I think that actually is a great transition for one of our other poll questions we have here,
nice.
Our next poll question.
So we're going to shoot up that poll question.
Yep.
And also we did send the link to the lab tour video in the chat. So people can copy that link and watch the lab tour after this session too. So how many cardiomyocytes, which are the heart muscle cells, do you think are lost as a result of a heart attack?
And as Martha said, this is pretty much the whole, or kind of one of the main motivations behind what we do is trying to make these particular cells to then put back into the heart. Looks like 40% of the people said 10 million cells, 20% 1 billion, 30 at 1 million, and 10% at 1,000. So the correct answer is actually the astonishing 1 billion cells, which is quite a few, which is why a lot of the work that, Martha mentioned a little bit earlier, is, you know, we can make, you know, you can make a small quantity in something. But one of the big things with especially the engineering aspect of trying to produce these cell therapies is how to make large batches of these cells.
So to give us some context, how, like in your lab today, how many cells might you have growing?
In one plate, which would be kind of about that size or so, for reference, or I guess about this maybe 2 iPhones worth, if you want to go that way. You'll probably have on the order of about 10 to 20 million with an M. And we're trying to make, you know, each patient needs 1 billion. So, I mean, you're hundreds, you know, an order of 100 larger than that. And one of the other things with cell therapies, especially in with cardiomyocytes, is we want to be able to produce not just one, enough for one patient. We want to be able to produce large batches that could potentially be used for hundreds of patients. So then you take that 1 billion, you multiply it by 100 patients, and you're looking at 100 billion cells you want to be able to produce. So that's well outside the scope of anything we can do in our lab right now, but a lot of the work we're trying to do is to try to improve the production process and the overall quality of the cell.
Evelyn has a question related to that. So you're working really hard every day on these really important questions and work. So what do you do if an experiment fails?
Yeah, it's when an experiment fails, not if an experiment fails. I will say, I mean, this is research. These are you know, new questions and things that not a lot of people are working on. So things fail all the time, and that's probably one of the less glorious sides of research that people don't talk about, or you know, people at your stage might not necessarily know. You know, I'd say, you know, when I'm doing a differentiation, so I'm taking my stem cells and trying to make them into cardiomyocytes. You know, I'd say maybe sometimes as much as half of my experiments will fail, or half of my differentiations will fail. So you just kind of have to shrug those off and go to the next one and keep it going. Sometimes you can get a little depressed, you know, if you hit a streak of several failures, but you kind of have to keep in mind that kind of final goal, you know, kind of the final goal at the end of the day, and you're working towards that goal, and kind of look back and see how far we've come. And then you can really boost yourself back up when you start getting, you know, some really good results, really interesting results. You can say, okay, well, I know I had many failures along the way, but it was all to get to this really interesting piece of information that's now going to, you know, change the world, for example. But it's a good question. Failure is everywhere in research.
So we've got some more fun poll questions. So maybe we'll send up that poll question. And a question about the heart, about how fast does the average human heart beat? And while we ask that question, I was wondering a question from Martha. How long does it take to grow a cardiomyocyte in the lab? And how long are you working on these cells, or maybe, you know, waiting for things to grow?
Yeah, so that's a very good question. So what we do is we start with stem cells, and then we differentiate. There's 3 different germ layers in your body. One of those is mesoderm, so we differentiate to mesoderm. And then after that, we go to cardiac lineages. That takes about 6 days to get to cardiac lineages, and then to cardiomyocytes, which is about 14 days total. So think 2 weeks. So every experiment is 2 weeks plus the assay. So we don't have data fast at all.
No.
And it's 2 weeks minimum to get to the minimum viable cell type. Some of the work we do, you know, you'll extend them. I think I've got cells in culture right now that have been there for almost 300 days now. So nearing a year that those cells have been in the incubator.
Yeah. And the poll question results came back. Most of the people think that the human heart beats about 60 beats per minute. And one person's very slow, 1 beat per minute. And 13% say very hummingbird-like 200 beats per minute, very fast. So Aaron, about how many beats per minute is your heart beating right now?
Right now I'd have to, you know, count it out, but it should be right around 60. 60? Probably a little bit higher than that. I think, you know, Olympic runners are down in, you know, the 40s and 45 resting heart rate. You know, an average person is typically in the 60 to 80 range.
Yeah. And Martha, can you describe what these heart muscle cells do in the lab? Are they beating, and do they look like a heart? And how do we tell?
Yeah, so this is something we showed you in the video, but the cardiomyocytes will actually contract. You can watch it under a microscope. The other thing we could use is a calcium dye, which we also showed in the video. So the way that works is when the cell beats, there's a calcium influx. And so then the cell will fluoresce, and you can watch the fluorescence over time. It looks like a strobe light.
Yep, and both those videos are in the YouTube link, so I encourage you to watch those. They're
Yes.
it's really neat whenever you differentiate these cells in the dish, and at the end you can actually see them physically beating on the plate. It's really reinvigorating every time.
Yes.
The question about how does it recapitulate the heart, I think, leads well into the next poll question. So in the dish, it's flat, right? But we want to know how many chambers are in the actual heart, or human heart, I should specify.
And like an adult, fully developed
Yeah.
heart.
Yep.
Cool.
I think even actually as early as the late third trimester, it's actually in its fully developed structure.
Yeah. And as people are answering that poll question, I'm going to ask each of you kind of a related question from Braylon. Braylon's wondering if you play any sports, or did you play any sports to get that heart rate up?
Yeah, I guess right now in COVID times, not so much. But so I used to, especially in Madison here, I played ultimate frisbee a decent amount, racquetball quite a bit, both of which are high aerobic sports for sure. Also, some older people sports I've been told people call them, golf, bowling, things like that. I've done that. Used to play baseball, soccer, football a little bit, although I'm really not built for it. So I've played kind of a number of sports for sure.
How about you, Martha?
I'd say I like being outdoors more. So I like to ski, I like to bike, a little bit of kayaking every once in a while, hiking, rock climbing, more outdoorsy kind of sports. If they're
That's awesome.
called sports, I don't know.
Yeah. And we have results from the poll question. Can you guys see that?
Yep, looks like most people are right at four chambers. So there's two ventricles, what are called ventricles, and two atria. I think there's a good figure on the YouTube link there, where it actually kind of shows what each of those does and where the heart's pumping blood from each of those different chambers. But yeah, 4 chambers in the human heart. And the mouse heart, a lot of mammalian hearts are 4 chambers. Other ones have simpler kind of 2 chamber, or even some very simple organisms have a 1 chamber heart. I think we have a little bit of time left. I'd like to get, if we can get to this one last question, or I guess this is the second to last question, but we can make it our last question here, which again kind of motivates some of the work that we do. So maybe that'll kind of bias your answer. But how fast do the cardiomyocytes, so in your heart right now, how fast do your heart cells regenerate or replicate? How often do they kind of reproduce, go through mitosis and make a new one, or make, 1 into 2?
And this is related to previous questions we had when you get a heart attack and your heart needs to repair itself, or as your heart's growing.
Okay. Like we've got a tie for 1% per day and 1% per month. The actual answer here, by the time you're about, I think, 12 or 13 years old, your heart cells, those cardiomyocytes specifically, really only generate about 1% per year. So if you think about that, that basically means, by the time you're 100 years old, your heart will have essentially turned over once from the time you were born. With those rates, although technically that number diminishes over time as well. So a lot of the cells that you were born with, a lot of the heart cells you were born with are still the exact same cells, and they haven't replicated since you were born. which is part of the reason we need to be able to make new ones if you damage them, because they can't regenerate themselves very well. It's a very, very slow growing cell, not like your skin or anything that, grows over really quickly.
So that's so important and exciting that you get to see these cardiomyocyte cells in the lab day to day. And we have got one last poll question. Maybe we could close out with that. Where do you see yourself, or what do you see yourself doing after high school? And as people are answering that poll question, Martha, maybe you could share with us one of the more favorite things that you get to do every day being a scientist.
Well, I think the cool thing is when you design an experiment and then it works. You get a result. You get something that's novel and new to the field. That's something that nobody else has ever discovered. That doesn't happen very often. That's like, once every 2 years maybe, but it's worth it.
And Aaron, does everyone here need to go and, like you both did, and get a PhD to be able to do science and research like this?
Not at all. So, I mean, so a lot of undergrads work in a lot of labs. Even shy of that, high schoolers, if they're particularly interested, there are definitely opportunities. You can reach out to, local, if there's particular people that you're interested in working with. A lot of even university professors are happy to welcome high schoolers into the lab to help with research. So there's a lot of avenues where you can get involved in research or in kind of the cell manufacturing field in general. You don't have to go all the way and get a PhD if you're interested, if you want to kind of have some of those experiences.
Yes.
Yeah, we worked with a student from the community college here. He got a 2 years associate's degree, and now he works in a company where they bank blood. So that's totally possible as another option of things you can do. You really don't need like a super degree.
Totally. And we've got a widespread in the audience here today about next steps in their lives and their careers in science. And it's all leading to really exciting areas in all kinds of science. So we're at our time. So I'd like to thank everyone for joining us here at the Wisconsin Science Festival. Thank you to our presenters, Martha and Aaron, for joining us from the University of Wisconsin-Madison. A recording of this field trip may be available, and we'll follow up with teachers about that. Remember, the Wisconsin Science Festival continues with events hosted from organizations all across the state that you can participate in today, this weekend, and beyond. Just check out wisconsinsciencefest.org with more fun science, and we'll see you there. And thank you, Martha and Aaron, for joining us.
Thanks for having us. Thanks, everyone, for joining, and stay safe and best of luck on your future endeavors.